I wouldn't forget to mention it to you in the morning. The question for today's discussion is a little bit different. You're gonna spend a little bit of time with your group coming up with an answer to the question. You're gonna do a little bit of discussion just as a whole group about it in class. And then you're just supposed to put on your unified answer as a group online by tonight, okay? Just a paragraph or a little video. You guys can make a little video out of it. You don't have to write anything. You could just have a conversation in a, like, do a Zoom recording of it or kind of studio recording and post that. It does not it doesn't have to be very prepared. I don't want it to be very prepared. How about that? I want it to be how you would sit down and discuss with each other. That's the way I want it to be, okay? I hope.
So you got the 104?
No.
Oh.
She was tired.
And then there's another kind of an assignment that's coming up. That's just for you guys to do on your own and then post and then kind of discuss each other's doings. What you're doing is playing a kind of a scientific game, using ideas that we've been talking about how we create different organisms, how organisms evolve over time, how they survive and adapt to different environments. So there are a few different options that you have available, whichever one is easiest for you and readily available to you. You don't have to choose one that you know, the first one, if that is harder for you to get to, just pick one and do that and then post. Okay, so this is the imagine that you can design a code for a robot using the same principles as those described in the chapter self-assembly for cell differentiation and organism development. Describe the main components of that system and how they would help you develop the robot code. And you want to make sure to talk about like advantages, disadvantages of that system that has been presented in that article, right, in that little chapter, and how it would affect your robots. Okay. So just keep that in mind. We'll talk about that in a little bit. So think about that as we go through today's lecture as well. So you can kind of, you'll get some help from that as well. Did everyone pick up their papers, Frank?
Yes, so.
If not, you might wanna. Okay, are there any burning questions about exam or anything else that you guys have been mulling over the last several days and want to ask? I know some of you talked to me and through Zoom over the break, but if you do, you can, again, come by my office hours. We can talk about any of the questions in the exam. Once I have everybody's graded, we can have a little bit of discussion like I did last time. Hey, so what are we going to talk about today? So we're going to talk about we're going to overview what we already kind of talked about with gene expression and how it happens. And then we're going to talk about several ways that it is going to be controlled, both before it ever gets started, right? So transcriptional regulation. And then we're going to be also talking about regulation at the level of translation and then post-translation. And so we are gonna then, towards the end, we're gonna be talking about how all this helps us, essentially develop specialized cells for each function. So first of all, with transcription regulation alone, how it helps us, and then later on, we'll come back and rotate towards it again after we do post-transcriptional controls and talk about how other parts of our systems are regulating this as well. So, why we need that? Well, first of all, we all know that we have the same genome, regardless of which cell type you're gonna look at, right? So as a multicellular organism, we have all these different cell types inside us, all different functions that are happening by each one of those different cell types. But when we look at inside the nucleus, at the genome, it's all gonna be the same, right? Every single cell has the same recipe book at the end of the day. But they're able to differentiate somehow into very, very different things. So what are some ways that it can do that, given that everyone has the same recipe book? What's different then? What is it that you are doing in order to get those different cell types? Well, all of that is because which one of those recipes are actually getting produced in a particular cell at the level of RNA and then at the level of proteins. Different types of cells are going to express only a subset of those recipes, not every single thing that's there. There are certain genes, there are certain proteins, right, that have to be produced no matter where you are. Those are what we call housekeeping genes. And then the remaining are specific to specific cell type. Whether you are a cell in the liver versus in the stomach or kidney, it's gonna dictate what type of things are going to work with it. So through that process, this is something that we have looked at, and we've looked at how differentiated cells are able to differentiate over the course of their development to become eventually a multicellular organism. There are a few experiments that I do want you guys to be in knowledge of and kind of know how they were done. So the first one is one that actually helped us figure out where exactly differentiation starts and how exactly this happens to begin with. And in this particular case, they took adult frogs, they took the skin cells from that frog, and they went ahead and they took just the nucleus from that adult frog cell, and they added it into a unfertilized frog egg where its own nucleus was destroyed by UV radiation. So they destroyed the egg's own genome, and they took this adult frog genome in the form of its nucleus and injected it into the egg. And when they produced an organism out of it, they got a completely normal frog eventually, right? They got a tadpole and a normal embryo, a tadpole and a frog. So what does that tell you, that particular experiment?
That frog was made.
That what?
The frog was made.
That a frog was made. And what does that tell you about the genome?
They have the same genetics.
So even though it was not a haploid nucleus from an egg or a sperm that had to come together, it was an already adult frog. It wasn't a gamete anymore, right? That the nucleus came from. It still contained all the instructions that you needed to create the eventual frog. So it wasn't anything different in there, right? From what was in the gametes. Another experiment they did was again looking similarly, but this time looking at a plant cell. They took a section of a carrot and they proliferated the cells that were present in there using liquid media. And once they had a mass of cells, they separated them and created, remember in class, we all create those single cell suspensions, right? Where we take the cells off the matrix they're sitting on, and then we create a kind of the suspension with cells in it that we can grow. So they did the same thing. They separated these cells in some liquid medium. So they had a suspension. They then took individual cells and they plated them to examine if they were able to create a fully functional plant, essentially. And what they found was they could get, just from a single cell, a clone where you would get a young plant eventually and a carrot, just like normal. Again, this also told you that, it didn't matter what type of cell you're starting with. If you have the right environment, you can actually keep them proliferating, get more cells, and if you give them in the right situation, they can develop into a fully grown plant that it came from or an animal that it came from. Again, showing that the genome is the same throughout that process. And the last one is where we've been looking at our cloning, right? And with the cloning, you have, again, the same process, but at a more macro level. In this case, you can take, again, cells from an adult cow, right? And it's the same idea as the frog, that you take an unfertilized egg, you destroy its own genomic information, and you replace it with genomes from an adult donor cell. Typically, it's from the epithelial cells because they're easier to proliferate and get and work with. And once they do that, they are able to actually reconstruct a zygote and reconstruct an embryo that can be produced into a clone calf, which will look just like the donor, not like the mom, even if the mom is different, right? So the genome is controlling the phenotype, obviously, so it's gonna match more the phenotype of the nucleus that it came from. So all of these essentially tell us that the genome was the same. It had all the instructions all the way up to the adulthood. But what was different was that you had to create the environment where they could now differentiate into another organism rather than just be the specified adult cell. It also tells you, because remember, we are just taking sections, right? We're taking it from the frog's skin. Similarly, we're taking it from an epithelial duct. So these are differentiated cells. These are not undifferentiated cells. So technically, they shouldn't be able to so-called create the entire organism. They should be giving you skin cells, but they don't. If you provide them with the right environment, you can de-differentiate them as well, not just differentiate them. So given the right environment, you can make these cells express different types of genes to do completely different function. So as we talked about, there are proteins that are common to all cells. These are called housekeeping proteins. They come from these housekeeping genes, and they're going to be necessary no matter where you are. For example, ribosomes. Every cell has to make ribosomes, right? Every cell has to make certain type of transcription factors, polymerase. There are things that are just going to be common. Every cell needs cytoskeletal structure. But then there are other proteins that are specific to the cell type that it is in. And those specialized proteins are what are going to be responsible for that cell's function at the end of the day. And this can happen through expression of genes, through internal changes in the expression of genes, through internal signals inside the organism itself, but they can also be put on it by signals given from external environment, right? So if you change the environment of that cell, you can end up getting a completely different organism. And for some of you that are interested in looking at the process of differentiation in the lab, you will find that that's exactly what we will do. We will have them in a certain environment normally to keep them from getting too differentiated in culture. And then when you add your agents to them and change their environment, they will differentiate into whatever it is that you were looking at, whether it's muscle cells, right, from myoblasts to myotubes, or whether they are your neuroblasts that are turning into neuron-like structures, okay? So gene expression needs to be regulated. Basically, that's the end of the game, that to differentiate things and to do the functions that each cell, specialized cells have to perform, they have to be able to regulate their gene expression. And they do it at many, many, many different levels. It's not just at the level of gene transcription, but all throughout the process. So you can have transcriptional control at the level when the DNA has to be transcribed into an RNA, right, to get your mRNA messages. You can then have it at RNA processing. Remember, we've already talked about some post-transcriptional changes that happen with the alternative splicing to make slightly different forms of different proteins, right? From the same mRNA, for example. So that's a kind of post-transcriptional control. So that's in the RNA processing, how much it is processed and what its half-life is and whether or not it's going to be spliced a particular way. Those are all examples of that part. Once it's transported outside, just that process is sometimes at a control as well, whether it's going to be transported outside the nucleus immediately, or whether it's going to wait until another signal comes through, asking it to be localized outside. So that's going to be your transport and localization control. The next level is going to be whether, what's the half-life of your mRNA, whether it's going to be degraded immediately, or whether it's going to hang around in the cell for a particular amount of time and just be translated all throughout that time until it is no longer needed. And then once you actually get into your translation itself, you have how active that translation is, how fast it is, what's the turnover rate, how many molecules of protein is it going to produce, right? So some have faster translation than other, and we'll see examples of why that might be the case today, a couple of them actually. Then once your protein is prepared, it doesn't mean it's active immediately, right? Sometimes it just hangs around and it needs a binding partner before it can be activated, or it needs another post-translational modification. Does anyone remember some examples of post-translational modifications? No? Anybody else? Somebody. Get a stick. Well, it's more over here than there, but sure, I'll take it for now. What are more common examples of post-translational modifications? Yeah.
Like the splicing and the capping.
Splicing is all the way over here with RNA processing. So I'm specifically talking about modifications, yes?
Phosphorylation.
Yes, good job. Phosphorylation is a big one, right? Another big one more. Yes, good job. The sugars. Those are two big ways that proteins are modified in order to become activated, right? So there are glycosylations that can control the binding partners and its activity, and there are phosphorylations that can activate or inactivate a protein. So those, again, are going to be your activity controls that you're going to contain. And then finally, you also have the half-life of a protein. Just like you had a half-life of the mRNA, you have a half-life of protein as well that controls whether it's going to be, how long it's going to be around in the cell and whether it's going to be there to be active or degraded over time. So those are all different ways that our gene expressions can be controlled at different steps. So the first one that we're going to be talking about is our first part there, which is the transcriptional regulation. So in transcriptional regulation, there are, again, several different ways that it can be controlled. The biggest one is obviously the regulations by different molecules, regulators, whether they're repressors or activators of transcription of a particular thing. And this is done through, again, those repetitive sequences. We've talked about some types of repetitive sequences already with the Tara box and some other promoter sequences. But now we're going to talk about a lot of more specific ones. So there are regulatory DNA sequences that are present all throughout the genome that your transcriptional regulators are going to bind to in order to either activate it or repress it. Then you have obviously regulation of the chromosome structure itself. Again, we've touched on that before, and we're going to build on that again today. This includes our heterochromatin euchromatin conformation, right? Remember the methylation on your genome that can make it closed structure or open structure in order to be transcribed or not. And then you have these post-transcriptional processing and control, which controls whether the mRNA is gonna get ready to be translated immediately or not. So let's talk about transcriptional regulators. Favorite. No, not my favorite. I have others that are more favorite, but still, this is also. So transcription regulators are basically are proteins typically, right? These are proteins that have specific motifs or small DNA sequence or small amino acid sequences that bind to specific parts of your genome. So they're going to match up with specific DNA sequences that they are going to be binding to, and they're going to be affecting that particular portion of the genome. So there are a variety of DNA binding motifs that are present all throughout our proteins, families. There are alpha helices, obviously, that's one of the big ones that can help the protein bind to the DNA sequences. There are also what we call zinc finger and leucine zipper. Zinc finger domains are proteins that have basically binding sites for zinc as in the way that they are controlled. And they are going to, that binding provides it with the right conformation to bind to DNA itself then. Another one is leucine zipper. These are usually these coiled protein structures that essentially can open and close under different environmental conditions, and they can wrap around the DNA and affect that area. And then you have these homeodomains. Now, homeodomains can be different depending on which protein you're looking at, on what kind of other proteins it's gonna bind to, or specific DNA sequences it's gonna bind to. So homeodomains contain specific areas that can bind to only specific sequences on DNA. For example, P53, which is a tumor suppressor and can also act as a transcription factor, that has a particular binding site throughout the genome. Those sites on the genome are called P53 binding domains that the homeodomain of your P53 is gonna bind to. When it binds to it, it's gonna activate or deactivate downstream genes, gene expression related to that site, okay? So most of these regulators bind and are activated as dimers. So them need to be together to be at full functionality. If you have just a single regulatory sequence, you will have one of those protein molecules bound to that sequence where it has that homeodomain for that sequence. But to get the full activation effect, you need repeated regulatory sequences to recruit two of them coming together as a dimer. And once it is there together as a dimer is when you get the full functionality and the full effect. So here is one of those sequences that is shown in the genome. This is actually a protein very important for differentiation into different cell types. It's called NANOG, and it has this particular sequence that it is going to be more related to. So these, the ones that are more bigger letters, they are always there, right? These two A's are always there. The first side can be T or a C. These two can be T's, two T's or two G's together. They're more likely to be T's than G's, but basically it has a sequence that it is going to preferentially bind to. And the closer it is to that sequence, the more strongly it binds to that sequence and has an effect on there. A lot of the transcriptional regulators will have sequences like that in genome that they are going to recognize and bind. So there are two different types of transcriptional regulators. Obviously, they can be ones that activate gene expression, and those are called inducers. And then there could be ones that turn off or deactivate gene expression, and those are called repressors. You can also have activators that are essentially turning on the genes as well. So you have repressors turning genes off, activators turning genes on. And inducers as well as co-repressors are going to be small molecules that are going to participate with the repressors and activators to help them along with whatever function they need to perform. So co-repressors will bind to repressors, inducers will bind to activators. So that's kind of just, exactly what this sounds like. I have to remember to bring a new one. Mine has been stolen and gone away.
Oh, what are the ones with the inducers and the one?
So inducers and co-repressors. Inducers will bind to activators and co-repressors will obviously bind to repressors and help them along. Inducers and co-repressors can't do function by themselves. They need to bind to activator and repressors. Activator and repressors can work on their own as well. The inducers and co-repressors help them along, either making them better or in some case are required. Okay, so now this means, that's one part of the story. The other part of the story is that depending on the binding partner that these activators and repressors bind, so depending on what co-repressor or what inducer is binding to your protein, that is your main regulator, it can have a completely different function. So that is a second level of control. So you have these MYC genes, right? MYC binding sites, these are called key boxes. These are responsible for proliferation control. And so you have proteins that are, there's a MAX protein, MXD protein, and MYC protein. If MYC binds with MAX, it's going to activate proliferation. But if instead you have MXD binds with MAX, again, at the same sequence, it is going to instead repress proliferation or inhibit proliferation and instead activate differentiation. So depending on the environment that your MAX protein is sitting in, right? If it's sitting in a cell that is actively proliferating, you would expect to find MYC bound to it. That's the gene that's going to be, protein that's going to be more around for them. And if you are putting that same MAX protein in an environment where the cell is actively differentiating, maybe in a muscle or a nerve cell, it's going to be found bound to MXD instead. When it binds to MXD, it inhibits MYC from binding to it. When it is, and for active proliferation, you usually see there is no MXD around in those cells. MXD is going to be specific for differentiation itself. So again, same protein that's always binding to this EBOX MAX, but depending on what binding partner, so what co-repressor or inducer it's binding to, it has completely different function. Does that make sense? Yes.
Max categorized as activator or repressor?
So it depends on what binding partner it's binding to. That's the thing, right? So it's the same exact protein, but it is involved in control of proliferation. But it can be an activator if it is binding to MYC. MYC is an activator, by the way. Yeah. MYC is considered an activator. And in that case, MAX is an inducer along with it, and MXD is a differentiation factor. So in essence, it's acting as a co-repressor. Okay. And they're both MXD and MYC bind to the same sequence in that EBOX. It's not like they're binding to different areas. They're binding to the same sequence. It's just that it's the environment of the cell that they are in. Whichever one is present more or whichever one is exclusively present is the one that's going to be doing the function that it needs to perform. And again, you can look at their structure, and both MYC and MAX actually share a lot of these similar structural proximity as well. They're both part of the leucine zipper family. And so all three of them essentially, but it again depends on which one is present and which one is binding, and it's going to control what function it performs. So now we're going to actually start by first looking at this gene transcriptional regulation, gene regulation in prokaryotes, and with two different examples. And then we'll talk a little bit about eukaryotic gene expression control, which is a lot more complex. So we'll kind of cover it in a little bit more overview form. But I'm going to give you two specific examples from a prokaryotic gene expression control because those can be followed a little bit more easily. So in prokaryotes, the genes, we already talked about this before, can sometimes, all the genes involved in a particular pathway or a particular metabolic pathway, for example, right? Or a particular control system are usually in a row, all next to each other in the same mRNA transcript, which is called an operon, right? So they are all under the same, in the same mRNA. They all have their own AUG start sites and stop sites, but they are going to be coming off of the same mRNA without the splicing and all that shenanigans. So each one of these genes will have its own little ribosome binding site, and they can all have different levels of expression coming off of it as far as translation is concerned. So the mRNA, the full length mRNA that is present, you will have the, all the AUGs on there. When the ribosomes bind, they will bind to all of these AUGs. And when they start to translate, they will translate at different rates depending on the amino acids that are part of it. And I'll show you an example of that in a second. So this allows that you can, it's in a way a very efficient system, right? So if I need more enzymes related to lactose metabolism, for example, or glucose metabolism, I can turn on that entire system with a single switch versus individually going and starting them separately. And that's pretty cool. And they're all under the same promoter regulator system that once activated, everything will get transcribed at the same time, even if they are translated at different rates later on. So one of the examples of those is this tryptophan operator or tryptophan cluster operon. Just like any other operon, the TRIP operon is under the control of a single promoter region. The promoter region has your -10, -35 sites, right? They're gonna have the Tata box and all that other stuff. But then in between those sites, they have an operator region. The operator region, you can think about that is where your activator or repressors are going to bind to activate that to regulate that entire cluster downstream. In the TRIP operon, you have these 5 different genes that are needed to be transcribed. Each one of them is going to code for a different enzyme responsible for tryptophan biosynthesis, and all of them needed in order to make tryptophan. So if you need tryptophan, you can't just make A, B, and C. You need to make all 5 of them in order to get the tryptophan to be made, synthesized by the cell itself and to be working properly. The mRNA that comes off contains the transcript that has all 5 genes transcribed on there, so all of them can be translated together. Now, here is the cool part about that. Again, like I said, you have the -10 sequence where you have your Tata box, you have the -35 promoter sequence, and in between you have what we call an operator sequence. The operator sequence is where a TRIP repressor binds. So this is the tryptophan repressor. It is inactive when there is no tryptophan around, right? So if there's no tryptophan around, this just runs around, playing around, does not bind to the actual sequence. And RNA polymerase is free to go bind to this promoter sequence region and go ahead and start transcribing the operon, the tryptophan cluster. Now, let's say there is a lot of tryptophan hanging around the cell, then those tryptophan bind to this repressor, activating it. That's how it activates it, it's smart. And once it is activated, it binds to the operator sequence. And when it binds to the operator sequence, the polymerase can't bind to it, so it can't transcribe the genes. Cool, so far? Okay, so now we'll look at it a little bit more closely and see what happens next. So this is just the first level of control in the tryptophan biosynthesis. It's a simple, if there is no tryptophan available, the repressor can bind, polymerase comes in and transcribes the gene so you can get more tryptophan. When there's a lot of tryptophan available, the tryptophan binds to the repressor, activates it, and it can bind to the operator sequence and shut down the transcription. The next level of control is that now the transcription is happening. You're getting the transcript. It's going to be that entire operon. In bacteria, if you remember from before, our transcription and translation are coupled, right? So as soon as the transcript starts to come off, you start to translate it as well. That's usually how it is in the bacterial system. Well, in the entire transcript, which contains all the 5 genes, there are different areas that can create hydrogen bonding with each other and create these double-stranded stem loop structures. So one of them is that this region 1 and 2, there are 4 different regions that can do it. 1 and 2 can bind to each other, 3 and 4 can bind to each other. This is one thing that can happen. Alternatively, 2 and 3 can also bind to each other as well. And so the way this happens is that there, in that beginning sequence, and I'm going to draw it over here because it's not clear over there. So you have your transcript, right? So this is your 5' cap, right? 3', and you have your 3 areas that 5 areas rather. A, B, C, D, E. These are your individual genes. Always you have your 5', what we call untranslated region, quote unquote, and your 3' untranslated region, right? So these are areas that are not part of that operon. They do not contain any of these 5 genes, so they are not part of what's going to get translated. That's the extra sequence that comes along with these transcripts. All transcripts will have some of it, some lots of it, some not so much. Now, within that sequence, in a different reading frame than the reading frame of this one. So remember, we have those, let's say T, A, B, C, G. So if this was your sequence, there are 2 AUGs you can see in here, this one and this one, right? But they're in different reading frames. So if I make it like this to get this one, I won't get the second AUG. So it's in a different reading frame from each other. So in this so-called untranslated region on this side, there is another AUG that is in a different alternate reading frame from all the others. And this one contains a small peptide called a leader peptide. So it can be translated. It's not that it can't be, right? It's a leader peptide. And so we call it UORF, or open reading frame. So it's an open reading frame. And the U designates that it's in the untranslated region as far as the rest of the operon is concerned. So that particular leader peptide contains 2 tryptophan codons right after each other, okay? So if that leader peptide is getting built a particular way, so let's say you get, there's lots of tryptophan present, so there's going to be lots of tRNA with tryptophan, charged tRNA with tryptophans present. That leader peptide is going to be made right away, right? As soon as you come in, the ribosome comes in, finds the start codon, it will get those tryptophans and it will start to build it. That means those 2-3 structure cannot form, right? And the 3-4 stem loop structure will form, and it will terminate transcription because it's going to be like, oh, there's plenty of tryptophan here. Why am I making more? Because it was able to cross this path. It creates the stem loop structure that stops the transcript. And so what you end up with is your leader peptide along with this stem loop structure. Alternatively, if you don't have a lot of charged tryptophan or tryptophan available, so you don't have a lot of charged tRNAs with tryptophan available, the ribosome is going to be stuck. Those codons not able to move, it's stalled, right? Because it went to that AUG, it got to the start codon, and now it's a tryptophan and there's no tryptophan around. This allows the different, the 2,3 structure to form instead of the 3,4. And because it forms this structure, it continues to create the transcript, and you get the full length transcript and you get the full length translation. You have the operon to work with. Yes.
The stem loop structures stop the.
The stem loop structure 2,3 is going to allow transcription to happen. The stem loop structure, the first one where it was at towards the end, the 3,4, that stops. This stops transcription, that lets it go on.
But that's is the stem loop structure just completely stopping the tryptophan codon? It's like the 2,3, I understand that transcribes.
So
after.
it's not the one stopping the tryptophan codon. There's no tryptophan around to go through with this. So because it allows, this ribosome is stalled, but it allows another ribosome to come through and bind to the actual AUG in the operator in the correct site and start translating that.
I'm just confused on like the function of the bonds between like, so it's creating that loop because the 2,3 are bonding together or.
So basically, so it's not the fact that there's a stem loop structure that's stopping there, it's where it is and how strong it is. And when it is at that end part of your transcript in this 3,4 region, it causes a stop that essentially drops off the ribosome. Whereas here, it's transient and it can be controlled by another ribosome coming in and starting translation.
Yeah, I'll just ask you later.
Okay. Like I said, it has a little bit of a playing game there, but yeah, we'll talk about that too, but in another form as well. But basically, if the leader peptide is created and that other, the 3,4 stem loop structure is made, you're not gonna get more transcript getting built and you're not gonna get tryptophan operon completely made so that you can get your actual proteins. If it is not, if it's stalled here, no leader peptide is made, only the transcript is built, and that can be then translated into the individual proteins. So that's one way that you can control gene expression is through these repressors that are controlled by the product of that operon itself, essentially, right? So it's kind of an example of a negative feedback loop. When you have enough of that product, you no longer need to activate that system anymore. Another way, obviously, is by looking at activators. In activators, you can have, again, it's going to be within that promoter operator site. You will have a binding site for the activator protein that's going to have your activator bound to it. Sometimes they're going to only be present at certain times, so they bind only at that time. Sometimes you have an inducer that's going to activate the activator so it can bind. In this case, we're showing that as this bound activator protein. That activator protein has to bind to this inducer before it can be activated and before it can recruit your RNA polymerase to then create your transcript. So we'll see an example from there next. Might stay here for a few seconds. So this is another operon in your prokaryotes. This is the LAC operon, and it is controlled by two signals. It's controlled by an activator and a repressor. So two different things are going to work together in order to regulate when your LAC operon is active and when it is inactive. So if you look again at your LAC operon, you're gonna have the LAC-Z genes that are going to be part of the this particular operon. That's what we're trying to transcribe. You are going to have your -10 and -35 promoter regions, right? You will have your binding site for your promoter right there in the center. And before that, in front of that, you have your operator sequence that is right at the start of transcription. And a little behind the promoter region, you have another regulatory sequence that is what we call the cap binding site. The cap binding site is actually going to be part of an activator sequence there. The operator is going to be for the repressor and the cap binding site is for an activator, okay? So if you have lots of glucose and lots of lactose in your system, in this particular case, in this E. coli will always, just like any other organisms, will use the glucose preferentially, right? So they're going to be using glucose, and lactose is not really needed for its metabolism, so it's just hanging out. So your operon is going to be off. That helps you metabolize the lactose. So operon is going to be just switched off. Now let's say that you have a lot of glucose and you don't have a lot of lactose in the system. You still have a lot of glucose, so your system can still preferentially use the glucose. However, because you don't have any lactose, you want to make sure that operator is switched off. So in those cases, you have a repressor that is bound to the operator sequence, preventing the metabolism of lactose, whatever lactose is available, by putting that repressor in the operator sequence. Now, when, on the other hand, you have low glucose and low lactose, now you have to start trying to figure out how to utilize our no lactose working with it. Well, as soon as the glucose gets low, right, the levels of glucose go below a certain threshold, it activates the cap activator protein because there is no more glucose to metabolize, so you'll have a lot of cyclic AMP around, and that cyclic AMP is going to bind to the cap activator, and it's going to now recruit it to that activator site. So at that cap binding site, that's where it is. However, the operon is still going to be switched off because there is no lactose, the lac repressor is still present and still keeping it shut off. Now, when you don't have glucose and you have CAMP, there's a lot of CAMP available, the cyclic AMP, only your cap activator is going to be activated at that time. And now if you have lactose in the system, the lac repressor can come off and your RNA polymerase can actually bind and create the mRNA associated with the LAC-Z gene. So both of them together are required to control when they're switched on and off. And you can see in most cases, if there's plenty of glucose around, it's going to be switched off no matter what. Whether if there is no or low glucose, but there is no lactose either, it's still going to be switched off because of the repressor. It's only when there is lactose present in the system and low glucose that you will have the full activation of the activator and removal of the lac repressor, and the operon can actually function. Yeah?
Yeah.
So in our research, we actually utilize this a lot when we are doing transfection or creating expression systems that we can control using this lac operon promoter. So we utilize the lac promoter in our genomes, the plasmids that we create, where we are inducing our gene expression. Let's say I have a gene of interest that I want to study in prokaryotes, or I want to make it in prokaryotes so I can isolate it and then study its function. Well, I can look at how well I was able to transfect that gene, and this is something you might be doing in genetics as well if you're taking genetics, where you are fusing the gene of your interest to the lac promoter. So it's gonna get activated the same way as a lac operon does. And what you get at the end of the day is we have it fused to the target gene. So only when a lac promoter is activated, you will get expression of this gene. And typically we have it under control of this IPTG, which is, again, an artificial way to activate it, but it's going to give you that blue-white cloning. When you see the blue-white cloning, that's how we are doing it, is through control with the lac promoter. We could do it under promoter of different genomes as well. So you can have a T7 promoter instead of the LAC promoter, and you can put that under the same control using the same kind of sequences. So when IPTG is present, it would induce the expression of that particular target gene, just like it does when lactose is present, it activates the gene of interest. So this is one of the ways that we utilize this in research. Okay, so any questions on prokaryotic gene expression before we move to eukaryotic gene expression? Okay, so in eukaryotes, the transcriptional regulators, again, they're the same way. You have activators, you have repressors, but they don't have to be right there next to the gene. They can be far away, like hundreds and thousands of base pair away. And it's because of the way that the genome is kind of folded and packaged inside the nucleus, that they can bring those promoters close to each other, those activators close to each other in order for gene transcription to happen. So eukaryotic activator proteins will bind to sites on the genome that are called the enhancers. And these are binding sites for activator proteins. They can be close to the Tata box, or they can be far away from the box and the start of gene expression. But when an activator binds to the region, it allows for that particular part of the genome to create this kind of loop structure where it brings them in close proximity to mediator proteins and the polymerase so that it can actually activate gene expression. So you had all those different transcription factors. Remember, we had the transcription factor 2B, 2C, all of those are sitting in here recruiting the RNA polymerase, but it's not going to work until the mediator protein comes in, which is going to be activated by the enhancer in order to actually activate the transcription itself to move forward. And that's as much of this as we're going to talk about, because that requires a lot more understanding of the different mediators and the different transcription factor. But we will talk about other aspects of gene regulation in eukaryotes. Another aspect of that is, again, with the help of your the way the DNA is packaged with the histones, right? So you have the DNA that is packaged along the histones, creating those nucleosomes, right? You have the core of nucleosome, you have the DNA wrapped around it. You have transcriptional regulators that are somewhere on that sequence. You have the Tata box downstream of it. So when these transcriptional regulators, usually these enhancers, bind to those regions, it causes chromatin remodeling to occur. So it causes that entire region between the enhancer and the Tata box to open up and be in a more open conformation to turn into euchromatin instead of that heterochromatin conformation. So this remodeled chromatin now allows for that Tata box to be visible in order for other proteins to come in, the general transcription factors to come in, the mediator to come in, and polymerase to come in. On the other hand, if you have the transcriptional regulator bound, but you also have an alternate modification in the form of histone acetyltransferase, that's another way that you can actually create a site that is amenable for transcription. Because if you remember, acetylation can also end up leaving you a more open conformation sometimes. And those have to be specific acetylations that happen along the genome. And those are going to be done by a particular protein called histone acetyltransferases. And those do exactly what they say they do. They transfer an acetyl group to a histone in order to create a more open conformation. So all these structures are kept in check by the way that our chromosomes are first packaged by the histones and then packaged inside the nucleus. So they're not just, our genome is not just kind of laying around with every which way inside the nucleus. It does have specific areas for each chromosome to be laying in and a particular conformation to be in. So there is design to the madness, even when it is an uncondensed chromosome. And so the chromosome will have these little loop structures happening all along. These loops are kept in place by what we call clamp proteins that are going to keep these loops exactly in the right conformation so that your enhancer and your Tata box of the genes that they regulate are in close proximity to each other and can activate each other or can work together to activate gene expression or repress gene expression. So here are examples of how that might happen. You have these enhancer sequences here that are interacting with the Tata box of those genes that they are controlling. Even though they're far away from each other, because of the loop structure, they can be close enough to have an. And here it is, an example where you can see all of them in check. And so you can have, again, like I said, you can have thousands of base pair in between each one of these sequences, right? So these are the spacer DNA. They're all on the same chromosome, but they can be very far away from each other. And each one of these sequences, regulatory DNA sequences, are, again, one of those transcription factors or one of those mediator proteins, these enhancers and repressors that are going to bind to the mediator, to bind to the general transcription factors and activate the actual start of transcription. It's not going to be just done by these transcription factors alone that we talked about initially. So how do these regulatory proteins bind? They are going to be these regulatory proteins are specific to different transcription factor. Each transcription factor has its own binding partners. So we talked about, for example, with the MAX and the MIC, right? And MAX versus MXD, that's an example of a regulatory protein with its alternate binding partners. So each transcription factor that we look at has its own set of binding partners they work with and that they bind to in order to control transcription. And each one of them can typically be both activators or repressors of gene expressions based on the binding partners that they are binding. They don't automatically have to bind another protein or DNA. They can actually bind to the mediator on RNA polymerase. It can bind to RNA polymerase directly, right? Or they can bind to the DNA, right? So any one of those interactions can result in regulation of the gene expression contained by that system. Okay, so we finish at 10:40, right? So I'm gonna stop here and we'll talk about our chromosome modifications next time. So we'll start from chromosome modification next time and talk about all the fun things related to methylation of DNA. And we're gonna go ahead and talk about our discussion question now. Oh, I really wanted to show a good thing. Tuesday.
Actually, I read it.
You guys don't have lab on your Tuesday with me anyway, so it's okay. It will be fresh in your mind.
Everything else.
Also, by the way, a lot of you people are choosing turmeric. Please come up with something other than turmeric. At least some of you. I can't have the entire class choosing turmeric when I have 15 different things to examine.
Yes.
I like chosen.
I just remember that. Where is the?
We're doing research and it's.
There. So, go ahead and take 5 minutes within your.
Answer the question.
Have you guys working in your group? Imagine code for a robot. What were things that you learned in that original self-assembly chapter that would help you do so?
Ecology. There's more math than there is ecology. Bro, calculus, bro.
Study, I was trying to read your,
Shit, what are we even gonna talk about it? I don't know what to talk about.
I just said like was just submitted as a group. Someone.
So, talk to me.
Goes in order. We've done numbers.
It's the same. It's five. It's them, bro. No.
Eric,
Say.
stop Eric.
Hey, three.
Yes, we're going.
Yeah.